Hi, my name is Margaret Donovan, and I'm a bioinformatics scientist at Seer. I'm super excited to share with you our work on deep, unbiased, and peptide-centric plasma proteomics to enable proteogenomic studies of NSCLC at scale. So comprehensive assessment of the human proteome has remained elusive due to multiple forms of a protein, each of which can actually serve distinct functions arising from alternative splicing, allelic variation, and protein post-translational modifications, or PTMs. Characterization of the variable protein forms, or actually you can call them proteoforms, will expand our understanding of the molecular mechanisms underlying disease. However, it's actually the identification of these variable forms requires unbiased protein coverage at sufficient scale. Scalable, deep, and unbiased proteomic studies have been impractical due to cumbersome and lengthy workflows required for complex samples like blood plasma. And so here, we demonstrate the power of Proteograph Product Suite in a proof of concept proteogenomic analysis of 80 healthy controls and 61 early stage non-small lung cancer samples, or NSCLC, to dissect differences between protein isoforms arising from alternative gene splicing, as well as the identification of novel peptides arising from allelic variation. So here's a study overview of our proteogenomic analysis of healthy controls and early NSCLC samples. So walking from the left to right, we have on the left sample collection. So we have obtained plasma from healthy individuals, early NSCLC, late NSCLC, and individuals that are identified to be comorbid. We applied two different LS-MS approaches, generating both DIA and DDA data. On the top, we outline, or actually sort of in the middle, we outline two different peptide analysis approaches. So from the DIA data, we performed a discordant peptide intensity search, which essentially meant that looking between the early NSCLC subjects and healthy, we identified differentially abundant peptides. And then mapping these peptides to genomic space, we asked if these differentially abundant peptides were associated with any known protein coding isoforms, and were thus able to identify proteoforms based on gene structure. And then the second approach we took was using the DDA data, for which we looked at early, late, NSCLC, healthy, and comorbid. And from these individuals, we actually had matched genomic data for which we generated custom databases. So using the genotype data, whole exome data to create custom FASTA files, and then which allowed us to make personalized databases, and then we could identify protein variants. And all these together, both the discordant peptide search and this proteogenomic approach has enabled us to identify many proteoforms, which is allowing us to sort of build out this space of what is sort of already captured and known, or at least has been searched for in the past in the database of plasma proteins. So addressing sort of that first approach here, using this discordant peptide search, we found that detecting peptides and not just proteins is critical to understanding biology. So after we applied that approach of identifying differentially abundant peptides, we identified a number of proteoforms. So here I'm actually going to walk you through an example of one of those we found, which is BMP1. So BMP1, we found that if we looked at it at the protein level, if we asked between healthy individuals and early NSCLC individuals, do we see a difference in BMP1 abundance? And the answer is no, we didn't see any difference, especially no significant difference. However, when we took this peptide-centric approach, so we looked at all the peptides mapping to BMP1 and looked at the differential abundance between healthy and early individuals, we actually found something quite striking, which was in early NSCLC samples, we found increased abundance of the first two peptides, which corresponded to you can see those first two exons. And then the final 5 peptides mapped to, actually the final 5 peptides were overabundant in healthy individuals. And if you put this, and you can see this in the bottom part of the central portion, you can actually ask and look at where are these peptides mapping with respect to these 4 known isoforms. And what we see is those 2 first overabundant peptides in the early cancer subjects actually correspond to a short isoform, and the overabundant peptides in the healthy individuals correspond to these 3 longer isoforms, which really told us that there actually may be an association between this short form and long form with health state. So maybe potentially the shorter form is associated with, maybe a lung cancer state, and these longer isoforms might be more protective, and that's why we identify them in the healthy individuals. So, really what this told us was that using this approach, not only are we finding and identifying, or we're enabling the inference of proteoforms, but these proteoforms are actually very meaningful for biology. So the second sort of part to this work is our proteogenomic search. So as I mentioned previously, we used healthy, early, late NSCLC and comorbid subjects, for which we generated proteograph-derived proteomic data. We also generated 100x whole exome sequencing data, which we called variants. In the genomic data, we did translation and in silico digestion, and then using that, we were able to create custom DBs. So these are peptide sequences that actually reflect genetic variation from these individuals. So that in combination with the reference database, we were able to put those together and make a personalized database for which we can do a variant peptide search. So across these 29 subjects, after doing this personalized proteogenomic search, we identified hundreds of peptide variants. You can see in more detail the number of variants we found, the number of peptide variants we found across these 29 individuals. And we also highlight an example where one individual had a heterozygous variant, for which in the mass spec data, we were able to identify both the reference and alternative peptide variant. So together, what our work shows is that hundreds of proteoforms can be inferred from proteograph data. A peptide-centric approach uniquely enables proteoform identification, and these identified proteoforms recapitulate known biology as well as capture new disease-associated biology. Together, this tells us that Proteograph can generate unbiased and deep plasma proteome profiles that enable identification of protein variants and peptides present in plasma at scale sufficient to enable population scale proteomic and proteogenomic studies. Thank you so much.